This will continue in the fall semester. And if you are interested in participating, then please reach out to me if you want to speak. We already have a couple of speakers, so that's really exciting. And yeah, I'm just going to introduce Amanda. So today's speaker is Amanda Furuyu. She's a PhD student in entomology under the guidance of Dr. Hugh Smith. Amanda received her bachelor's degree in agronomic engineering from the Sao Paulo State University in Brazil and her master's degree at the University of Florida. Her area of study is integrated pest management focused on alternative management, and she's currently working on Diamondback moth and Brassica crops. In addition to her responsibilities as a graduate student and researcher, Amanda serves as the GCPSA secretary, and so she'll be taking the helm of GCPSA leadership next year as our 2024 GCPSA president. And with that, Amanda.
Okay. Can everybody hear me? Yeah. Thank you for the kind introduction, Elisa. Good morning, everyone. My name is Amanda Furuyu. I'm a PhD student under Dr. Hugh Smith, as Elisa already did the introduction. So today I'll be talking about the influence of Brassica variety on density and parasitism of Diamondback moth larvae and pupae in Florida. This is not working. So, a lot of technical issues today. So just to give a brief overview, Diamondback moth is a key pest of the Brassicaceae family, including weeds in some important varieties. Diamondback moth, or DBM, as I am going to be referring to it in this presentation, is a cosmopolitan insect, and the damage is caused due to the larval feeding, as you can see here in this picture. It leaves this window pane aspect on the underside of the leaves, and the annual cost of DBM control can reach up to $4 to $5 billion. So DBM is a brassica specialist, including many commercial varieties such as cabbage, broccoli, cauliflower, kale, collards, and so on. And some Florida growers reach up to us and they seem to be observing less DBM numbers in some varieties than the others. So for example, they think that they see less DBM numbers in red cabbage compared to other type of brassicas. So how true is that? Do you think like maybe DBM actually have these preferences to some brassicas than other brassicas? So by looking at these pictures, we can see that they have different features. So these brassicas, they have differences in regarding texture, colors, architecture. So for example, the red cabbage and the green cabbage, they have this architecture where the leaves overlap each other, forming this closed head in the middle of the plant. Collards, on the other hand, they have this configuration of the leaves that are more open. And Napa cabbage is something intermediate. They start open, they start enclosing with time. So with these different brassicas, these differences affect the number of DBM incidents. And in addition to that, we know that there's some natural parasitoids out there. So could these brassicas, these different brassicas, also affect the parasitism of DBM? So here we try to determine this veracity of these observations. So we did a survey to collect background information, not really focused on management, but to better understand the relationship, the interaction of DBM and host plants and parasitoids in actual field conditions. The objectives of our studies were to compare DBM incidence and parasitism among green cabbage, red cabbage, Napa cabbage, and collards, and also to compare DBM incidence in parasitism among 3 different brassica production regions in Florida. So moving on to the materials and methods, this trial took place last year in spring 2022. We look at 4 different commercial brassicas: the red cabbage, cultivar Red Jewel, green cabbage, cultivar Bravo, the Napa cabbage, cultivar Chinese Pacifico, and the collards, cultivar Top Bunch. They were all grown without any application of insecticides through the whole season, so the insects could come. And we The trials took place in 3 distinct Brassica production regions in Florida, represented by the red stars in the map. So one of them was placed in Hastings, up in Hastings in the northeast part of Florida, another one here in Baum in the southwest part of Florida, and another one down in Belle Glade in the southeast part of Florida. For these trials, we have 4 host plant treatments with 4 repetitions each in a randomized complete block design. So each repetition was represented by a plot with 20 plants each. And the data was collected at 3, 6, 9 weeks after transplanting by sampling 2 infested plants per plot. So the most infested plants per plot were the ones collected. So each repetition would be represented by 2 plants. So all these plants, they were collected and they were brought back to our lab. We look at each plant leaf by leaf, collecting all the larvae and pupae that we could find. And they were kept for further rearing so we could assess parasitism. All those DBM that we collected were kept in petri dishes like this. So since we had several individuals, like hundreds of individuals, it was not feasible to keep them and rear them individually in each petri dish. So each one of these have hundreds or a lot of them. And so we observe them daily. We feed them with fresh leaves offered according to the host plants that they were collected from. And then we waited until the emergence of either DBM adults or parasitoid adults, and they were all quantified. To assess parasitism, once the adults emerge, the parasitoid adults emerge, they were collected, quantified, sorted by species, and put into vials with ethanol for preservation. So here in the pictures, we can see this is actually a DBM pupa that is parasitized by parasitoid. So it's the parasitoid cocoon inside the DBM pupa. And the DBM larvae or the parasitoid larvae trying to coming out of the DBM larvae, and then the adults coming out of the pupa, the DBM pupa. So continuing, to estimate the number of DBM parasitoids, we did a proportion of 1:1, meaning that 1 DBM will be parasitized by 1 parasitoid when they were solitary. But when they were gregarious parasitoids, which means that like a lot of parasitoids are coming out of one individual, it's a little tricky. So what we had to do is that we got the species, the specific species. So in this case, our gregarious parasitoid, spoiler alert, is Omyza sokolovskii. So we had to look into the literature, and the proportion that we use based on the literature is that for 1 DBM, there will be around 8.4 parasitoids coming out of it. So for a more accurate estimation, they need to, the DBMs or any insects, they need to be reared individually in the petri dishes. But anyways, continuing with the methodology, for the species identification, all those samples, like my parasitoid samples, they were sent to the DPI, the Department of Plant Industry up in Gainesville. They did the PCR analysis, sequencing the gene CO1 of the parasitoids, and then matching those to the gene bank or both for confirmation of the species. For the data analysis, general linear mix model analysis was used to characterize the effects of host plant, site, and sampling time on the variables plant weight, DBM numbers, and DBM parasitism. The experimental design was considered a split plot when we analyze it, and we treated site as the whole plot factor and the host plant treatment as the subplot factor. Also, the plant weight and DBM abundance data was subjected to log transformation, and the percent parasitism was subjected to arcsine square root transformation for the uniformity of the data. Being that, the analysis was performed using the Glimpse procedure using the software SAS, and the pairwise treatment differences, the Alice mean statement was used with lines option. Moving on to the results, we're gonna start looking at each one of the effects. We have a lot of them, but for time's sake, we're gonna only focus on the site, host plant, and the interaction between site and host plants. So starting with the plant weight of the plants, we can see that there was a significant difference among all the sites and among the host plants, but there was no interaction between site and host plants. So here in the first graph, here on the left, we have the, I think my pointer is not working. Yeah. Oh, so here in the, so here in the vertical axis, we have the plant weight in grams. Here we can see how they change it among the collection sites. So what we did here is that we pull all the other effects to try to focus in one effect. So here in the collection side, we pull all the host plant types together. So we can see that in overall, the plants are larger in the southwest part of Florida, followed by northeast and southeast part of Florida. And when we look at the across all the sites, regarding host plant type, Napa cabbage are the largest consistently among all the sites comparing to the other host plants. Moving on to the DBM infestation, we can see that there was a significant difference among the sites, among the host plants, and there was an interaction between sites and host plant. So here we have the total number of DBM per plant on the vertical axis. Here in the first graph on the left side, we can see how the DBM numbers change throughout the season. So as expected, the DBM numbers, they increase with time. Here in the middle graph, we can see how they vary according to collection site. So there was a higher number, a significant higher number of DBM in the southeast part of Florida, followed by southwest, and then the lowest numbers in the northeast part of Florida. And here in the last graph in the right side, we can see how they vary according to the host plant type. So there is a higher number. They all separate, and there was a significant difference among all of the host plant types. So we can see that there is a higher number in green cabbage, followed by collards, and lowest in red cabbage and the lowest in Napa cabbage. Like we heard from the growers that they were seeing less numbers in the red cabbage. So because the plants are different sizes and weight, we also wanted to compare the infestation or the total DBM numbers using a consistent unit of measure. So we look at the DBM number per weight basis. So here we can see that when we look at that, there was a significant difference among sites, host plants, and the interaction between site and host plants. When we look at the graph, and when here we have the DBM numbers per gram of plant on the vertical axis. When I look at the numbers per weight basis, we can see that there's actually no difference between green cabbage and collard. So they have similar proportions. And red cabbage is still has lower numbers, and Napa cabbage has the lowest numbers. This is not what we expected because since Napa cabbage is the biggest plant, we thought that there would be more resources or biomass, and we'll see a larger number, a higher number of DBM on Napa cabbage. So now switching gears to DBM parasitism, this is the list of parasitoids that we got. These pictures were taken by Natalie, which also helped me with the molecular work. She took these amazing pictures of my parasitoids. If you can see here on the top, this is 0.5 millimeters. So this is really, really small. Well, continuing, so this is the list of parasitoids that we got in our survey. This one is the Diamondback, which is one of the most common parasitoids of Diamondback moth. It parasitizes the larval stage of DBM. It has a solitary behavior, which means that one parasitoid comes out of one individual of DBM. It was found in all 3 sites, and it was found in all the 4 host plants, the green, red, and Napa cabbage, and the collards. This next one is Microplitis plutelli. This is also a larval and solitary parasitoid. It was only found in the southwest and southeast part of Florida, so we did not find it in Hastings, for example, and we could also find it in all the 4 host plants. Next one, Cotija vestalis. This is also a larval solitary parasitoid. This one was only found in the southeast part of Florida and also in all the 4 host plants. Next one is the Sokolovsky, which I already mentioned it. This is a larval pupa parasitoid with the gregarious behavior, which means that a bunch of them can come out of one individual. This one was only found in the southeast part of Florida, and we also could find and collect them from all the 4 host plants. This one is one of the smallest parasitoids that I've seen in my life. This is the Acerofronidae species. This is a hyperparasitoid of the Diadema solari, which means that it's a parasitoid of the parasitoid of Diamondback moth. This one it has a gregarious behavior. We found it in all the 3 sites, but only in Napa cabbage. We didn't find in any other host plants. Next one is the Cornuta torvina. For me, this is the coolest one. This is a pupal parasitoid with a solitary behavior. We only found it in the northeast part of Florida and only in the collards. And we also found a Pteromalid species in the southwest part of Florida, only in Napa, and also another species here that we couldn't get to the species, but it's an Olophidae. It's from the family Olophidae, and we only found it in the southeast part of Florida and only in the green cabbage. So as for the effects on the parasitism, we can see that there was a significant difference among the sites. There was a significance on host plant at 0.06 and interaction between site and host plant at 0.08. So here in the first graph, we can see the percent parasitism in the vertical axis. As for the DBM numbers, the parasitism percentage also increased with time. Here in the middle graph, we can see that according to the collection site, there was a higher parasitism, a higher percent parasitism in the northeast part of Florida comparing to the other sites. And when we look at the host plant type, we can see the percent parasitism was higher in the Napa cabbage comparing to all the other host plant types. So now moving on towards the end of the presentation, regarding the conclusions for DBM infestation, we can see that DBM infestation increases with time, which is expected. The highest numbers of DBM infestation were found in southeast part of Florida, followed by southwest, and lowest in the northeast part of Florida. And higher numbers were seen in the green cabbage, followed by collards and red cabbage, and the lowest in Napa cabbage. Even though the Napa cabbage is the heaviest, is the larger plant comparing to the other host plants, when we look at the DBM numbers on a plant weight basis, we can see the Napa cabbage actually has the lower numbers, and the higher numbers are in the green cabbage and collards at the same proportions. As for percent parasitism also increased with time. Percent parasitism was the highest in the northeast part of Florida and in Napa cabbage. And we also saw some differences regarding parasitoid species. So for example, there were more species in the southeast part of Florida and more species in the Napa cabbage. The species of Sokolovskii and the Cotegia vestalis were only found in the southeast part of Florida. Micropletis plutelli were only found in the southwest and southeast part of Florida, and the hyperparasitoid Serafronidae was only found in the Napa cabbage type of host plant. So reaching the end of my presentation, I would just like to acknowledge the USDA crop protection and pest management competitive grants for funding my research, my PI, Dr. Hugh Smith, Dr. Julian Bosley in his lab, Dr. Bill Turchak, Dr. Talamas, Natalie, and Matt from DPI, and all my lab members for all the support. Seriously, this would not happen without them because it's really hard to count all the DBM with crickets and cockroaches jumping on you. So you guys are the best. Thank you so much. And sorry for that. But yeah, that's it. Thank you so much for listening. And I'll take any questions now. No questions? I would just tell my, professor first. Okay, ladies first. Hi, Cherry.
I'm just curious because the the different cultivars that you use for different are those the widely grown cultivars here in Florida?
So the question that Cherry is asking me is if I chose these varieties because they are the most grown in Florida, and I'll say yes. That's why we decided to go with those four. Those are the most important brassicas produced in this region in Florida, and so that's why we decided to choose those ones.
Just a follow-up.
Yeah, sure.
Because I'm just curious, from there, if you, if you chose a different cultivar, do you think there will be an effect in, DBM infestation for each of those different, cabbages?
So the follow-up question is if I had used another cultivar, if that would affect the results for that specific variety. Is that right?
Like, for example, if you chose different cultivars.
For
for
red
different
cabbage.
impact,
Mm-hmm.
do you think variation infestation, or do you think it will be most likely the same thing?
That's a really hard question to answer.
I'm just curious.
Yeah, because this is we try to see what's happening out there in actual field conditions, but it's really hard to tell what is affecting them. So I don't know if we try to use the most common cultivars, but I don't know if like just changing cultivars will actually change the major features of that specific variety. I don't know if that answers your question. Yeah, okay, I try my best. Please.
The professor is the last. No, it's just I was just wondering if you did or you plan to do any research, like experimental research regarding the characteristic of the brassicas. I was wondering, for example, if one of them has more wax, would it
Yeah.
be harder to think infection or something like that.
So that's why I'm starting my PhD. That's why I went for a PhD. Yeah, well, we discussed this. It's really hard to try to tell. Oh, sorry, I forgot to repeat the question. So Lee's question was if I have a follow-up project or research to actually see the different features of the different brassicas. This is really hard to tell. It's really hard to measure like the waxiness, or I don't know, quantify the different, if the real different architecture of them really affects them. So we're trying to think on a way of actually measure that and try to evaluate the consequences on the DBM behavior. That's really hard, but yeah, that's in mind for sure. Alex.
Mine,
Oh.
mine was building off of that.
Uh-huh.
They've seen in like pomegranate, for example, they see differences in or associations with higher anthocyanin content or metabolism.
Right.
So that was my question. If you were looking at that.
Yeah, I thought about that because they have different concentrations of glucosinolates. So brassicas, they have this glucosinolates that actually is kind of toxic for insects, and DBM are specific. They have this specialization. Oh, sorry. Specialization to break down those chemicals and be able to feed on them. And like these different brassicas, they release different amounts of all these glucosinolates and isocellulates and all of them. I don't know if I thought about it, I discussed it with you already, if there was a way to actually quantify the amount of the metabolites, because that's something that gives a cue for them to come and overdeposit in those plants or not. So yeah, that's a really good question. Mhm. Right. There's a lot of factors. There's a chemical factor, that could be a more morphological factor, and all of those things. Like I need to, yeah, my next chapter. Please again.
Sorry, I was just like thinking, like after your answer, like some insects, they are attracted more for one color to other. That's why we have different threads too. And as you show, like the varieties has different colors. If that would be like one,
I don't know if the color would actually influence because they're all in the same spectrum, but I think that would be more related to either morphological or chemical cues. That's my opinion. like so far, like based on my observations of a master scientist. Yeah. Team.
Thanks so much for the seminar. This Diamondback seems to be a big play on the end, so it's good that you tackle that. So I'm wondering how much there is already out there in the research about, you know, different interactions, molecular, chemical level between cabbage or brassica and diamondback moth. And if there's not, can you think of an organism where you can maybe adapt some methodologies?
That's a really good question. You know, one of the things that I was thinking, when you're trying to think like how to quantify those metabolites, what is the easiest way? Like, should I put like other insects with them so they release the actual amount of metabolites and that's gonna like attract all of those dB? I don't know how to quantify that. I don't really know how like plan a methodology to do that, because there's no much in the literature about it. There's something about the difference concentrations or the proportions of the metabolites for those brassicas, but not really how to quantify those as a consequence of insect feeding, for example, for this diamondback moth. And if that amount of chemicals is something that would attract or affect the feeding preference or oviposition preference. So this is something that is really hard to tell right now. But yes, I don't know if that answers your question.
I don't want to go into detail too
Yeah.
much, but we can chat about this later.
This is my first semester in my PhD, so go easy on me, please. Mhm.
Maybe, but think about aphids and
Right.
viruses. I think there's been research out there about fecundity and the
Right. Right.
And then think about like closed environments where you can actually measure some, is it gas chromatography or something where you can measure, or
Mhm.
think of the research that has been done with those sensors in the field where you actually have a microchip that detects different volatiles, you know. So I believe just think everything and then pick what's close by and practical. So there are devices out there that could measure things like that, I believe.
Yeah.
So maybe.
Thank you for that suggestion. That's really great. If my boss wants to spend some money, right?
Or collaboration. Yeah. So maybe remember aphids as a model organism.
Right.
Methodology, volatiles and all that. Sounds super interesting.
Yeah.
It's cool that there's nothing really done that much in that. So
Thank you for that. Dr. Smith.
So we did this in Belgrade here in Hastings, and we found overall that there were differences overall in plant size, right? Does that mean that on a commercial level, you think that there's a difference in those three regions in what growers produce? That's kind of one way that can be that what you presented can be interpreted.
Right.
But I think we just need to be careful about it because Belgrade and Hastings were bare ground. This is on the classic mulch.
Yeah, that's one of the things. I didn't show it here. So the way that I presented the data, it kind of seems to look like that there was differences according to the region, exactly like the south part, central or north part. But there's a lot of differences among all the sides. The temperature, we didn't record the climate, we didn't record the weather. There's the weather. The weather is different. You know that like in South Florida is different from up Florida. The soil, the type of soil is also different. In the south part, they have muck. Here is more sandy, and Hastings is a little bit different. And also, usually brassicas, they are grown in bare ground. And here in this region, especially here in our center, everything is grown on plastic. So there's also that factor. So we can like for sure say that the differences in all the sites is because of the places these places in Florida, because there's a lot of things, other factors that are influencing. Any other questions?
I have a question.
Hi, Ellen.
Okay, thank you for that interesting presentation. It was really, really informative. Just to build on Dr. Smith's question a little bit, you say that you did not measure all these other factors that might be contributing to the differences that you saw. So I'm wondering how much of the observed variation did what you used to measure that difference actually contribute to?
Can you repeat your question? I couldn't.
Okay, let me rephrase. So you say that you did not measure a lot of components regarding the environment,
Right.
but based on let me say the cabbage type, how much does cabbage type really drive the differences that you saw? Like by how much? Do you have a value? In other words, how much variance does cabbage value explain to kind of give us the whole picture?
That's a really good question. I wouldn't know how to say that. Like just looking at the host plants, like how much that's the weighs on the results that we got. Is that what you're asking?
Yes, in other words, how much variance does the variables that you measured to come up with that conclusion, like how much variance do they explain, because we're missing some parts of the picture? So I'm just wondering how much does it explain, because we're missing a couple of important components. So yeah.
Can I get back to that question later, Alan?
Yeah, sure, no problem. Yeah.
Okay, thank you. Team.
Can you tell me a little bit more about how cabbage is grown, how those brassicas are grown, but maybe how a Diamondback moth has been controlled, Maybe I missed that in your presentation, but is it?
I did not went over that. So
What's been done? What are people doing now?
So the main control for DBM is chemical control. So pesticides, chemical pesticides. There's no much for biological control for DBM, mostly BT products and some fungi, but the parasitoids, for example, they're not releasing mass or anything. There are some on the market, but it's not one of the main methods to control DBM. So the main control method is still chemical control. So that's why DBM has already been recorded to be resistant to more than 99 active ingredients, because there's no rotation. I don't know, they have this short generation that changes quickly. So they develop resistance to many insecticides pretty quickly. So yeah, but the main go ahead.
Follow-up question, host resistance. I forgot my question, but that's what I remember, I mean, I'll think about it.
Yeah, we can talk about it later. Yeah. If it comes back to you, you can talk to me later. Hi, Dr. Smith.
Because that's why we're studying it.
Right.
Globally, Diamondback is like #2 of pests likely to develop resistance. So that's why you study it. And in fact, there are a lot of naturally occurring parasitoids that can help when growers aren't spraying a lot, but they spray a lot. So, and what Amanda's doing is pretty unique. People aren't really looking into the background. These aren't released parasitoids. It's just part of the environment. So,
and parasitoids, or you don't grow cabbage in the greenhouse setting or in controlled environments, or it's all pretty much field setting.
Field setting, yeah.
But my initial question was, cabbage has not been utilized so much for in breeding programs. So mapping a resistant gene has not been done yet. I suppose.
There are very few examples of post-plant resistance for managing archaeans. So yeah, so that's not part of, but kind of what Amanda's trying to get at is there are what are some of the characteristics that might be reducing it under actual growing conditions?
Okay. Thank you very much. Okay. Sorry, too late. Oh, sorry, guys. Yeah, the QR code for my speaker evaluation, please. Be kind to me.